distance.
Thank you very much. Thank you, organizers Jasmine and Dominic, for inviting me for this virtual talk. And hello, everybody, and especially for those who were there in the first workshop that I met in person. So I will be talking about some concepts that already were mentioned several times in this conference, but I also have chosen this topic to show, especially the younger participants, how valuable are those workshops. So the project that I will be talking about, if I can move the slide, okay, initiated a long time ago in one of these kind of workshops that was organized in the Institute for Mathematics and its Applications in Minneapolis, was organized by Tracy Jackson, that some of you met in the previous workshop, and Amy Radunskaya. And in my group was Judith Perez Velasquez, who is spending now the whole semester at the Fields Institute, who was a postdoc at that time, Jana Geverts, who was giving a talk in the last workshop, Alexandria Volkening, who was a graduate student at that time, Karian Norton, postdoc, and Sara Minzare, who was a graduate student at that time with the Eduardo Zontag group, who is in the workshop with you as well. So we work on this workshop, like I think the format was maybe closer to the previous workshop that you guys have. We work on the project, we make the presentation, and this time the organizers make the special issues of the proceedings from this meeting. So we published one paper in these proceedings, but then we continue working in different configurations from this in this group and published 3 more papers, one of them at the beginning of this year. So I would also include this in this presentation. And all those papers were using the agent-based modeling, but then Jana Geverts branched, used continuous models, and I think you've heard some of the results in, again, Eduardo Zontag talk. They have several papers on those topics as well. So from this one workshop, more than 10 years ago, we really have a lot of results over years. So the main keywords or key aspects that I want to talk in these presentations is drug-induced resistance, the tumor microenvironment, and how we could use modeling to identify some of those ecological niches. So you've heard about the drug resistance in this workshop a lot. So for us, this is when the tumor stop responding to the treatment, to the chemotherapy in our case. And you've heard that the biologists can generate resistant cell lines, and then there are 2 different forms of how the resistant can be generated, the pre-existing and acquired. So we are interested in the acquired resistance, where all the cells at the beginning are sensitive and the resistance will be developing due to the drug that was used in this treatment. And our question was, can this arise because of the microenvironmental heterogeneities? So when the resistant cells are generated in lab in vitro, the cells are exposed to the same concentration of drug all the time. We were interested what would happen if they are exposed to different concentrations of drug at different time points. So in order to address that, we have to generate the heterogeneous microenvironment. So we decided this agent-based model, so we have a patch of tissue. We created 4 irregularly spaced vessels. So if you model the oxygen diffusion from these vessels, you have irregular gradient. We model also the drug, which is applied from those vessels. It may have different diffusion coefficients, so the gradient may be not exactly the same pattern, but has a similar trend. And then we will be looking at the cells that will be growing in this microenvironment, and we want to trace how much the cells are absorbing the drug, so that's the different shades of pink, and how close they are to death. So it's something that we call viability. So the green is they are far away from death, and then the darker colors will show that they will be close to dying because of the drug. But we started with a small colony, so we also wanted to trace what are all the offsprings from each of those cells. And we call this clones of origin because they are not real clones. All the cells are identical at the beginning, but then we can trace their offsprings. So mathematically, the mechanical part of this model, we use the off-lattice model. So our cells are represented by the nucleus and the radius. They will be relocated due to some forces that act of them, and the relocation is inversely proportional to the viscosity of the medium. And the forces will arise from the cell overlaps. So we don't want the cells to overlap, so we will apply repulsive forces to push them apart once they divide. And then we have two continuous equations for the oxygen and for drug gradient. Both of them diffuse, they are taken up by the cells, they are supplied from the vasculature, and then we have decay for the drug. And because we have cells off lattice and then drug on lattice, then we have to introduce the cell neighborhood. So the action between those two entities will be only within the radius of the cell, so in the local neighborhood. So with these mechanical equations, we don't have to define how the cells will respond to the microenvironment. So first to the oxygen, they will uptake the oxygen, and then depending whether they are in the well oxygenated areas or hypoxic areas, they will divide or not. So in well oxygenated areas, they could divide. In hypoxic, they will just try to survive and will not divide. And how the cells will respond to the drug. So we consider a hypothetical drug that will induce cell damage, and the cells will have some small tolerance to the damage. They will try to continuously repair damage, but on the very small rate. When they are exposed to the drug will induce damage, and if the accumulated damage is higher than the cell can tolerate, the cell will die. However, if the cell is exposed to the drug, low level of drug for the prolonged time, then they become more tolerant to the drug, sorry. So the tolerance will increase. And upon division, we assume that the daughter cells will inherit both the damage and the level of tolerance. So this is illustrated in those three lines here. So I'm showing you on the population level how the tolerance can increase. The black line, how the damage, the blue line, there is no damage at the beginning, but then due to the drug, the damage can increase. It could exceed the threshold for some cells, and then cells will die. But if the damage will become divergent from the tolerance, that's what we have the drug that is the cells as a population will not respond to the drug, so become resistant. But those are like two different values. So we actually, in the whole presentation, I will be talking to you about this viability curve, which is the difference between those two. So if this viability curve increases, that's what we will decide that the tumor is resistant. So this is just the summary of what we have in the model and the whole flow chart. And let me show you how this model works in action. So in the simulation at the moment, you will see how the tumor is growing, and at the end, we will see this curve. So there will be growth of the tumor. Then once the drug has its action, a lot of cells will die, and then finally they will regrow and keep the constant population, which will be resistant. And you see these different colors, so the cells are all viable at the beginning, and then once they grow and accumulate damage due to drug, they become more dark. Once they are really black, they will die. But then with time, there is some population that survive and then regrow the whole tumor. And once they regrow and keep the constant population, their viability is also changing. So at the end of the simulation, all of those cells have high viability. So their damage is far away from the tolerance to the damage. And that's what we called the resistant tumor. So this is all on the population level. Oh, this is just the summary. And I was showing you only the colors for the viability of the cells, but we of course are tracing those clones of origin. So when we start with 64 cells, at the end, we end up with 15 clones of origin. All the other were eradicated. So this is on the population level, but then we got interested and see, okay, so what happened on the individual cell level? That individual cell became resistant. Oh, sorry, I go ahead. So the first question that we were asking ourselves, is this resistance built in the model or not? So we did some control simulations, and we can have 4 different cases. Depending of how fast this tolerance to damage grows, we can have tumor eradication. So all the cells were dead. We can have small residual tumor left. We can have the resistant that I just show you, and then we can also have non-responsive tumor. Then the damage curve never reaches the tolerance level. And those are a couple of cases that I just told you. Okay, so with that, we were interested what happened on the individual level. So we wanted to draw these viability trajectories for each individual cell. So to do that, we have to go from the final simulations to the first. So we look at all the cells that survive, calculate their viability, and then go back in time to the mother cell and grandmother cell until we reach one of these 65 initial cells and draw these viability curves. So as you see, of course, all the cells are viable at the beginning, and then for different cells, the situation is different. There are some that have this initial maximal viability. There are some that have this prolonged minimum viability. And then there is a steady increase of viability, but different cells, this has like a different shape. So we were intrigued by this shape and look what are the different subpopulations that we can see in this cumulative picture. So we identified 3 different subpopulations. The green one is the case when the viability grows really fast at some point. We call this like a rainbow-like viability. The red one is more linear growth of the viability. There are some fluctuations, but on average it's linear. And then we find out those blue, which are kind of like in between. Sometimes they try to grow like the rainbow-like, but then they go back and then they grow more like the linear. Each of this population has also different pattern of drug uptake. So the green one actually at the very end of the simulation, their uptake diminishes, sometimes almost to 0. So they don't uptake much drug. The red one have the constant uptake, and that's the equation that we put into the model, the constant uptake of the drug. And the blue are somewhere like in between. Sometimes it diminishes, sometimes come back, and is constant. So when we look at this, our model have space. So when we look at these subpopulations in space, they were actually nicely use different space in our different locations. And I come back to that, where those green and red cells are located here. But that's what we observed. So with that, we see here on individual cell how those different viability curves are different, but we would like to also trace for each cell individually all those information in space. So we've chosen the 12 cells that have the same origin. But as you see, there are 3 cells of each of those kinds. So 3 green rainbow-like, 3 red linear, and then the blue, which are in between. So we want to trace this in space. So what I will show you in the simulation is how the drug gradient will be changing. That's the background here. There is no drug at the beginning. And then with time, you will see the different pattern of the drug. And then there is one cell here, the mother cell, that will give rise to those 12 cells that we are looking at. So we can trace this in time and in space, where those cells started, when they're divided, when they ended up. And like in the previous case, so the green one are in those regions, there is far from the vasculature, red are in the regions maybe closer to the vasculature, and blue are somewhere in between. They started from the single cell that was dividing, and you see at the beginning there is a lot of relocation. We don't have migration in our model, but this relocation is due to pushing by the other growing cells. But then with time and the end of the later stage of the simulation, they are more or less parallel. So that means that there is tumor cells around that keep those cells in the same space. There's some wiggling due to proliferation of other cells, but they are pretty stationary. So that's again like still pictures from the same simulation. So the next question that we ask is, we know that all those individual cells become resistant, the viability curves are steadily increasing. Can we pinpoint where they became resistant? They were all sensitive at the beginning, so at which point they became resistant? So for that, we start looking at the lineage trees. And you've seen a lot of lineage trees today. It's the same concept. We have the precursor cell on the top, binary tree when the cell is dividing. Maybe one difference from some of the trees that were shown today is that we also show when the cells are dying here. So we have some broken branches. And we'll be interested in those subtrees for the cells that survive to the end of the simulation. So since we have these 15 clones, at the end of the simulation, we can look at each of those clones separately. So this is one that was not too big, cell number 4. And we are interested in those cells for which at least one successor survived to the end of the simulations. And this we will call the lineage tree of survivors. And if we draw this tree, this is the full tree. It's not binary anymore, because if one of the branch died, that we are all drawing them on the same branch, but we can observe how those trees of survivals look like. And we can do that for all of the subclones. If we look closer, and then lineage tree for cell number 26, I was showing you those spatiotemporal roots. So for those that survive, it looks similar like the movie that I show you, but we can also look at all the cells, and you see this kind of like a camp coming here of all those cells that either die or left the domain, right? There is a lot of, if the cells are proliferating, they may push the cells outside of the domain, which are then removed. So the full tree, the spatiotemporal roads have all those cells that were dying, but the ones that survive are more clean here. So once we know which cells survive, can we actually identify those cells that we call the precursors of resistance? So that would be the cell that all their survivors survived, all their successors survived. So we want to identify cells in this tree that have the full subtree. So the cells either survive to the end, or they could leave the domain. If the cell leaves the domain, we don't know if it will die or not, so we treat them as survivors as well. So those cells are shown here with the red arrows. They produce the full tree that survived to the end of the simulation. So once we have identified those cells, now we would like to know where they are in the domain. We know that they arise at different times here, right, from this tree, but we want to also know where they are in space. So first we look at the time. So they arrived in different times, and we broadly divided this in those three classes. So there are some early precursors of survival, that they are still on these viability curves that are far from the minimum. They are those magenta cells, they are in this prolonged minimum on the viability graph, and then there are those cyan cells that arise in the later times. So they arrived in different times where they are in space. So if we put them in space, and this is like the cumulative space, right? So it's not at the same time, they arise in different times, but when we project them in the space, they are actually localized as well. So those green cells are in the areas which are far from the vasculature, when there is a low level of drug, and those we call the pharmacological sanctuaries. This term was actually used before we use it by other groups. So those will be the areas when the regions are depleted of drug. The cells are sitting in the low level of drug, and in our case, that's the area when they will then increase the tolerance to the drug. The pink one are actually in the areas that potentially can have high concentration of drugs, but we think that they are protected by the other cells around, so that there are cells around that absorb more drug, and then those cells are then protected. That's what we call the cellular protectorate. So the cell can have time maybe to repair the damage, or because of the other cells absorb the drug, they are in the lower levels of drug, and then they can increase their tolerance to the damage. And then the third area is the cyan area, which is mostly near the hypoxia regions, that again, we think the cell are, the level of oxygen, the level of drug is not so high here, so they have time to repair the damage. There is this one region near the vessel that was published like that, but I think that should be like the fourth population here, because those cells satisfied our definition that they are precursors, that they generate the tree that survive the whole simulations, but those arise really late in the simulation. So sometimes there is only single cell, they don't proliferate and survive to the end of the simulation. So with that, let me summarize before I show you the last slide from our last paper. So what we show with the simulation is that it's possible that the homogeneous colony, but exposed to the heterogeneous microenvironment, so in this case, drug gradient, can acquire resistance. We show in the simulation that there are three routes of the cell adaptation, the rainbow-like, the almost linear, and then like in the intermediate viability pattern. We developed the method to identify where the resistance can be initiated, and that was done by first creating the full lineage tree, then the subtree of survivors, and then identify the precursors of resistance. And that led us to identify those three ecological niches. The sanctuaries, when there is cell have less damage and they can increase tolerance. Those near hypoxia regions, that they may have time to repair damage due to the low level of exposure. And then the protectorates, when the nearby cell may absorb the drug, and then the cell in this protectorates can then increase their tolerance to the drug, to the damage induced by the drug. So because we were using all those three-dimensional lineage trees in our research, we thought that may be a good resource for others. So we think about that like the extension of those Mueller graphs that were also shown in this workshop. We just add the space to those graphs. So this was published in the beginning of this year, and it's available on GitHub. If anybody will want to try, we provide the routines to draw the whole lineage tree, the tree of only surviving clones and individual clone in both of those configurations. The reviewers actually made us to make this in the different languages. So we have a Python version that was made by Kayode Olumoyin, a postdoc in my group, the R version that was made by Morik Ojwang, my previous postdoc, and I was working on the MATLAB version of this code. And then if you have the data from the experiments, so the locations of the or the traces of the cells, we can also use the routines to draw those traces as well. So I mentioned the people who was working on that during my talk, but let me repeat that. So the whole project was initiated with the WAM collaborative team during the workshop at the Institute for Mathematics and its Applications. And then we continue working on that. And my current and past postdocs were working on the routines that I presented at the end. So with that, thank you very much. And if you have questions, I'll be happy to answer.